Today we're going to take a look at acetylcholine. I'll see you inside. So the reason I kind of want to talk about acetylcholine is because not only is it involved in just about everything, including your memory and your muscle movements, but our dietary habits can actually alter the levels of choline that we produce. And as you can tell, I already slipped up on my words because I'm getting ahead of myself. So acetylcholine is actually made out of a molecule that we get in our food called choline. And if we take a look at the foods that are actually going to be highest in choline, these are going to be things like whole eggs are probably the highest, fish, specifically fatty fish. As terrible as it is to hear, organ meats are another great source. And then we're going to be looking at other animal proteins like chicken and beef. So to put some numbers around it, 4 large eggs is going to get you your daily value, and just 5 ounces of beef liver is going to get you your daily value. So now that you know that, let's see if I can convince you to eat liver every once in a while in order to hit your choline requirement. So the first step is actually getting this choline into the brain. So choline is actually going to enter through the brain through what's called the blood-brain barrier. And this is a membrane that surrounds the majority of our brain that actually functions to keep out most of the things in our bloodstream. And what happens is there's actually going to be a lot more choline outside of our brain than inside of our brain. And one thing to know about membranes is they try and have an equal distribution on both sides. So typically we will eat choline and it will be absorbed in the small intestine. And now sometimes this choline can actually be picked up by what's called the choline transporter. And this is the specific transporter that will help get choline into the brain through this blood-brain barrier. So the choline gets into the brain through this transporter, and this same transporter is located on neurons, so choline can go right into the neurons. And now the production of acetylcholine occurs directly in the neuron with a specific enzyme called the choline acetyltransferase enzyme. And now I actually find this rather interesting because while I talked about choline, I didn't really talk about acetyl-CoA. So there are some things that may actually limit our production of acetyl-CoA and its availability inside of a neuron. So acetyl-CoA can actually be produced when we're breaking down glucose, fatty acids, and amino acids. And now let's say there's an issue getting these things inside of the brain. And the one that immediately comes to mind is insulin resistance. If for some reason our insulin signaling isn't working properly, we oftentimes won't be able to get much glucose into the brain. And now I can't say this for certain, but there's definitely a rationale for how this could actually reduce our production of acetylcholine. And the other thing that I thought of was the production of both cholesterol and ketones. These molecules are actually produced when there's an excess of acetyl-CoA. However, that doesn't necessarily mean there's an excess in our body. Typically, this will occur in the liver, which will start the production of ketones or cholesterol. But let's say we're doing something like an extended fast, where the reason that these molecules are building up is not because we are intaking a sufficient amount of glucose and other things. Rather, it's because we're actually catabolizing our tissue in order to produce energy. And then in this case, all of that acetyl-CoA will likely just be shunted to produce more ketones. And this could potentially lead to a reduction in acetylcholine production inside of the brain. And now all of this is skeptical, but it's simply just something to think about and kind of understand the complexities about sometimes you may be robbing Peter to pay Paul in terms of using specific nutrients in one area of the body where you're depriving them in other areas. That being said, this is probably a 1 in a million or even 1 in a billion phenomenon, and likely would only occur if you have some sort of genetic mutation that makes you prone to something like this. But it's kind of fun to think about. But getting back to acetylcholine, we actually store acetylcholine in vesicles inside of a neuron, and these are simply just like packages filled with acetylcholine. And there's estimated to be about 1,000 times more acetylcholine inside of these vesicles than there is just kind of floating around in the neuron. And the reason for this is so that when this vesicle actually releases the acetylcholine, there's a lot of acetylcholine secreted into the synapse. And the synapse is just the area between two neurons that allows them to communicate to one another. And inside the neuron, there are these little vesicles with little proteins on their membrane called vesicular acetylcholine transporters. And as the name might imply, these transporters are going to take the acetylcholine that's floating around and pump it into the vesicle. So now we have these vesicles stored with acetylcholine. What is actually the impetus to release it into the synapse to let it do all of its functions? So just like any other neurotransmitter, we actually need some sort of stimulus to cause what's referred to as an action potential. And we'll talk more about this later when we actually talk about the function of acetylcholine. But all that really needs to happen is that we increase the voltage enough to open what are referred to as voltage-gated calcium channels. And then calcium will actually allow this vesicle to bind to the membrane and release all of this stored acetylcholine. And this process is actually typically caused by other neurotransmitters, such as glutamate. And another neurotransmitter that could cause it is something like norepinephrine. And these are referred to as excitatory neurotransmitters because they will bind to their receptors on a neuron and then cause this neuron to release whatever it's got stored inside. So let's say you're sitting in school and you hear the fire alarm go off. You'll probably get a surge in production of glutamate and norepinephrine. Norepinephrine is also known as noradrenaline. And you'll have the production of a bunch of other neurotransmitters, but we'll just focus on these for right now. And then these neurotransmitters will bind to a receptor on the neurons that contain acetylcholine. And then this neuron will release the stored acetylcholine, and now we can take a look at what acetylcholine does when it binds to all of its receptors around the body. So acetylcholine can actually work in all sorts of precarious ways. Unfortunately, for the sake of complication, there are actually 6 types of acetylcholine receptors. They fall into 2 different categories. One is called the nicotinic acetylcholine receptor, and the other category is the muscarinic acetylcholine receptor. And there are 5 different muscarinic acetylcholine receptors, but they're just called M1, M2, M3, M4, and M5. And now, why are they called nicotinic and muscarinic? It was actually because they found that nicotine and muscarine actually stimulate these receptors. And you may know nicotine is in things like cigarettes, and there are actually now nicotine lozenges, and the effects of these lozenges are actually to improve cognition and work output. Which makes sense, because if they're stimulating the acetylcholine receptors, as you'll find out in a moment, acetylcholine is very important for cognition and memory. And if you didn't know what muscarine was, don't worry, because neither did I. But it appears to be what's called an alkaloid, which is simply just a molecule that's derived from plants, and it's actually associated with toxicity of poisonous mushrooms. And we can presume that the toxicity is due to overstimulation of acetylcholine. So we'll first get the nicotinic receptor out of the way. So these receptors are going to be found in the central nervous system, which is going to consist of the brain and the spinal cord, specifically in an area of the brain called the hippocampus, the cerebral cortex, and the cerebellum. There are also acetylcholine receptors, specifically the nicotinic receptors, on the heart, lungs, and in the immune system. And very specifically, there are acetylcholine receptors at what is referred to as the neuromuscular junction. And this is a small space that is between a neuron and a muscle fiber. And this is actually how the brain can send a signal to a muscle fiber in order to have it contract or relax. So now, how exactly is acetylcholine going to do this? Acetylcholine specifically will cause muscle contraction, at least through the nicotinic receptor. So these receptors are actually bound to a specific protein that allows different what are called ions into and out of a cell. So typically, in its resting state, this protein is actually going to be closed. Acetylcholine allows it to open, and then these ions will float through. And don't worry, because these ions are things that you've heard of. They include sodium, potassium, and calcium. In order to not stay constantly active, when a cell is not doing anything, it will be at a charge of around negative 70 millivolts. And in order to get it to become active, we need to increase the charge, or the voltage. Sodium, potassium, and calcium are all positively charged ions. And when you hear ion, you can kind of think of it just as molecule. And now there's lots of calcium and sodium outside of a cell, and there's lots of potassium inside the cell. And these ions are always trying to make it so that there's an equal amount both inside and outside. And now the only way that these ions can actually get into the cell is through these ion channels. And the only way these ion channels open up is when acetylcholine binds. So now because there's more sodium outside of the cell than inside, sodium will rush into the cell. And since there's more potassium inside the cell than outside the cell, potassium will try to leave. And I know what you might be thinking. If they're both positively charged, if potassium is leaving, won't the voltage of the cell stay the same? Well, sort of. So because the voltage inside of the cell is lower than outside of the cell, there's actually an attraction of positive molecules into the cell. It's like this electrochemical pull that's trying to pull positive molecules into the cell in order to even out the voltage. And since potassium is positively charged, there's this kind of secondary force actually pulling it back into the cell. So what happens is that more sodium enters the cell than potassium can leave, so the voltage of the cell will actually increase. And technically this wouldn't do anything unless it actually caused what are called voltage-gated calcium channels to open. And since we've increased the voltage, we have passed whatever voltage is required for these channels to open, and now calcium will actually rush into the cell. And now calcium is kind of like the boss in town. So if you have ever wondered how a muscle contracts, it's actually by calcium binding to a specific protein called troponin. And now I may have to do an entirely separate podcast on this because it's kind of a complicated process, but all you have to remember is that this actually causes a change in the shape of the muscle cell and allows for what are called actin and myosin filaments to form what's called a cross bridge and actually shorten or contract the muscle. So now that we kind of understand what happens, what are the actual effects of this? So if we look at the heart, this will actually increase the rate that the heart is contracting, and it will actually increase the contraction of our blood vessels. So this will increase our heart rate and our blood pressure. In the muscles, obviously, it will make the muscles contract. And now I did mention that these receptors do work in the brain, and this is because when calcium is in the brain, it actually allows, once again, for this release of more neurotransmitters. So the same way that glutamate was able to kickstart the release of acetylcholine, acetylcholine can actually kickstart the release of glutamate and dopamine. And I mentioned these two specifically because glutamate will be very important for our cognitive function acutely, and dopamine will be very important for our attention, motivation, and memory. So I really want to highlight dopamine because dopamine is actually vital for learning about what behaviors and actions will be worth investing energy into in the future. So if acetylcholine binds to what are referred to as dopaminergic neurons, and these are just the neurons that will release dopamine, it will transiently increase our production of dopamine, and whatever behaviors we are currently engaging in will be remembered in the future as being rewarding and beneficial. And while this certainly is not the whole story here, this may actually be one of the reasons for the phenomenon of learning to enjoy exercise or learning to enjoy a specific behavior. So let's say you didn't enjoy exercise when you first did it, but during exercise you release acetylcholine in order to activate all of your motor functions, and this actually downstream causes an increase in the production of dopamine. As long as there aren't other factors that would be reducing your dopamine production, you may actually create a psychological association with exercise and the perception of reward, making you more likely to invest energy into exercising in the future. With that being said, we need to think about this. This is probably why things like nicotine are so addictive. Not because it's stimulating acetylcholine, but because acetylcholine definitely has downstream effects to increase our dopamine levels. Therefore, we will associate nicotine with having a positive investment value and be more likely to invest in the behavior of consuming nicotine in the future. Now, the other thing that I want to mention that this nicotinic receptor will do is will increase what are called glandular secretions. And this would actually be something like increasing our glucagon production in the pancreas, and actually increasing the salivary glands' production of saliva. With that being said, I could probably do an entire podcast on each one of these receptors, so I'm going to have to move on to the muscarinic receptors. So the one important thing to remember from the nicotinic receptors is nicotinic receptors will act to excite cells, so turn on the function of cells. It will increase muscle contractions and increase learning and memory. So we now have to move on to the muscarinic receptors, so we can start at the M1 receptor. So muscarinic receptors are called G-protein coupled receptors. And now I've talked about these types of receptors a lot in the past because this is how most of the neurotransmitters and hormones actually signal. So G-protein receptors sit on the outside of a cell, and attached to this G-protein receptor is what's called a G-stimulatory or a G-inhibitory protein. As the name might imply, an inhibitory protein would inhibit the functioning of a cell, and a stimulatory protein would probably stimulate certain processes inside of the cell. So the M1 is a G-stimulatory protein, and when acetylcholine binds to the receptor, what will happen is that there's a specific protein inside of the cell that will be released. And this will actually turn on a specific enzyme called adenylyl cyclase, which will actually cause ATP to be converted into cyclic AMP. And then this cyclic AMP can actually activate a specific protein called pKa. And if you remember from anything in the past, pKa is the big guy in town. So there's lots of different machinery and enzymes inside of cells, but they're not always active or they're not always inhibited. Some enzymes actually take energy to turn off their functioning, and other enzymes take energy to turn on their functioning. And the way we do this is through a specific molecule inside of the cell called phosphate. And simply, we just go around attaching these phosphates, and it acts as the energy to either inhibit or stimulate a specific enzyme or protein. And what pKa does is it just goes around and adds all these phosphates. Specifically, we'll add phosphates to what are called transcription factors. And these are actually little pieces of protein that bind to the DNA and cause the transcription of other proteins. And this really is most of the time how certain hormones will affect the cell. Simply by causing new proteins that we weren't previously creating to begin to be created, or to shut them off for that matter. And there's actually a ton of different proteins that can be upregulated, but I do just want to mention two of them. One of them is called the NOS enzyme, and this enzyme actually increases the production of nitric oxide, which acts as what's referred to as a vasodilator, or simply allowing the blood vessel to expand. And the other is an aquaporin, which actually allows water to be transported into and out of a cell. And you can probably imagine all the ways that this can help us during exercise and even for our memory and cognition. If we're vasodilating, we'll have better blood flow. And you can think about these aquaporin channels as if you ever have gone to the gym and gotten what is typically called a pump or an expansion in the size of a cell, this could be due to the increased production of these aquaporin channels allowing more water into the cell. And now the other thing that this cyclic AMP does is it activates another enzyme called phospholipase C. And this will actually go mess around with a molecule on the membrane of the cell called PIP2. And it will actually turn PIP2 into two new molecules called inositol and diacylglycerol. And the inositol can actually bind to what's called the endoplasmic reticulum, and this is kind of like the storage unit for calcium inside of the cell. So there's calcium inside of the cell, but we need this inositol to actually bind to the endoplasmic reticulum to allow for the release. And luckily, this inositol will do just that. And now in the muscle cell, once again, this will cause the muscle to contract. But in the neuron, this will cause the neuron to be excited and release the neurotransmitters. So I was going to do it individually, but since this podcast is getting somewhat long, G1, G3, and G5 are all G-stimulatory receptors. But since they're located at different locations all around the body, they do slightly different things. But since you do understand the mechanism of action, we can kind of just look at what happens. So if we look at the M1 receptor, there are actually M1 receptors on what are called cholinergic neurons. And these are actually the neurons that will release acetylcholine. So acetylcholine can actually increase the release of more acetylcholine. In terms of the M1 receptor, it actually works to decrease our heart rate, which is interesting because on the heart, it actually transcribes for specific proteins that are called potassium channels, which will be embedded into the membrane, and these potassium channels allow the excess potassium inside of the cell to leave the cell. But since this time we're not bringing in any sodium, this is simply going to reduce the charge of the cell even more. And then the heart doesn't contract as often, so we decrease the heart rate. There are actually M1 receptors on the salivary glands, which will increase salivation, and there are M1 receptors on the sweat glands, which will increase sweating. Which definitely does make sense because if we think about those aquaporin channels, both of these require the movement of water. We actually increase bladder contraction, meaning we are less likely to use the restroom, which does make sense because when we produce acetylcholine, we are typically in some sort of movement, and that's not the best time for a bathroom break. With that being said, these M1 receptors can actually increase bowel movements, and this is because it causes the contraction of the bowel. Acetylcholine is being produced all the time, and acetylcholine can be produced when we actually have time to use the restroom. And in this scenario, it would aid in increasing our bowel movements. As mentioned, these M1 receptors are located on the pancreas, where they can increase the production of things like insulin and glucagon. And it also causes the dilation of blood vessels, which can improve blood flow during exercise, but also decrease our blood pressure. And then we mentioned the ways that it can increase the production of neurotransmitters that could aid in learning and memory. And our stimulation of the M3 receptor actually seems to increase the heart rate through the mechanism of increasing calcium levels, causing the heart to contract. But it's also been found to stimulate what are called glandular secretions, which are actually things like digestive enzymes that can aid in the digestion of food. And then the M5 receptor is a newer receptor, and it's not very well studied, but it seems to do just about the same things as M1 and M3. So these are the stimulatory pathways of acetylcholine, but it also has inhibitory pathways. And this is through the M2 and M4 receptors, which are called G inhibitory receptors. And these ones are actually pretty simple. So instead of starting that whole intracellular cascade, what these things do is they actually turn off that initial enzyme, that adenylyl cyclase enzyme, that will take ATP and turn it into cyclic AMP. So eventually the cell returns to more of a resting state. I actually want to take a step back for the moment because I mentioned earlier that M1 receptors will decrease heart rate, and I actually just made a mistake in my notes. The M1 receptor acts just like the M3 and M5 receptors and will increase the heart rate. It is the inhibitory M2 and M4 receptors that will actually decrease the heart rate. And now this one is even more complex. So G proteins actually have different subunits. So what happens is that one subunit will work on decreasing levels of cyclic AMP, while the other one actually works on opening up what are called G protein coupled inwardly rectifying potassium channels, or GERK channels. And this actually reduces the activity of the cell even more because it allows potassium to actually leave the cell, decreasing the voltage even more. And now mostly the M2 and the M4 are going to work on the heart to decrease our heart rate. However, interestingly, they also have receptors in the brain, and this can do a multitude of different things. So sometimes this can simply just work to inhibit further release of acetylcholine. But what's really interesting is these receptors can oftentimes be used for targets of analgesics or numbing medications. And this is actually because they can inhibit the neurons in our brain that will produce specific neurotransmitters and neurochemicals that signal for pain. With that being said, it certainly does seem like there's more research that needs to be done on this topic. Alright, I want to leave this one here for today because I already know it was long and fairly complex. And to be quite honest, my brain hurts a little bit. And I'm really sick and tired of hearing myself talk. However, maybe you kind of learned a few things and now understand the functions of acetylcholine. And maybe I even persuaded you to add a few organ meats to your diet. Or at least a few egg yolks. So thank you for listening. I hope you enjoyed, and I hope to see you next episode. Have a great day.